So far, pretty much everything we've covered in the podcast has been preparation for this episode. We've discussed a lot about DNA, genes, and biobricks, but today we're talking about the most important step for many projects, perfecting powerful protein procedures. I hope you liked that alliteration. Anyway, to make this episode as easy to understand as possible, make sure that you're caught up with the previous episodes in the series. Alright, let's get into it. In this episode, we're going to be going through a lot of technical terms. Here's a few of them: lysate, supernatant, pellet, denaturing, elution, and OD600. We're going to define all of these terms, so don't worry if you hear them and you don't immediately know what they mean. Today's episode is very information dense, so I have a couple of tips before we start. Of course, it's best if you are caught up with the series, but if you're not entirely caught up, listen for words that you do know to explain words that you don't know. For example, if I say the electrostatic coulombic attractions, and you don't know what coulombic means, you can infer that it has something to do with electricity, or rather charges, and attractions, as in pulling closer. Sometimes, even just using parts of a single word can help. For example, if you know either macro or molecule, then you can figure out that macromolecules are a large something, or some type of molecule. Both are helpful. Oftentimes, it's enough to get the gist of what a word means to get the more important message behind it. If you don't know how to spell it, just give it your best shot in Google and add biology or protein after it. However, I'm going to go through a couple less technical terms right now to get everyone on the same page. First, when we say media, we don't mean it in the everyday sense. The media is what you grow or culture your bacteria on. It contains the nutrients it needs to grow, as well as other compounds you may choose. Second, aerobic respiration will come up in this episode. In simple terms, this is the cell's process for producing energy in the presence of oxygen. Third, binary fission. This is simply the process in which a cell, which is almost always bacterial, splits into two, allowing it to duplicate. Aerobic respiration and binary fission are not a huge part of this episode, but I recommend checking out Khan Academy or the Amoeba Sisters episodes on those, if you're interested. Finally, it's worth mentioning that if we say pH, we're just referring to something's acidity. We won't get into specifics, but know that amino acids have different acidities, and that affects both their shapes and interactions with other molecules. Now, let's start after we've transformed our bacteria with our plasmid. The next step is to culture the bacteria, usually in high volume so that we can get a lot of protein out and have a lot available for downstream experiments. Here's Ellie Campbell to tell us more about culturing your bacteria.
So to do this, first you inoculate some kind of nutrient-rich media like LB, TB, or a strain-specific broth with an overnight bacterial culture and an antibiotic. And the purpose of this antibiotic is to confer selectivity. So when you create this very nutrient-rich environment, you're essentially inviting all sorts of bacteria to grow in there. But you don't want this because this can crowd out your bacteria that are actually expressing your protein of interest. So you add the antibiotic to kill off all bacteria that haven't been given this plasmid that contains not only your protein-coding gene, but also an antibiotic resistance gene. And that gene allows those specific bacteria to survive the antibiotic toxic conditions, whereas other bacteria that don't have this resistance gene will die off. So once you've inoculated your media, you incubate it, which usually involves shaking to introduce oxygen into the culture, which supports aerobic respiration and metabolism, and you'll keep it at usually a warm temperature. So in the past, I've used 37 degrees Celsius, which is about human body temperature. Bacterial growth is a sensitive process, and it typically consists of several phases that are very distinct and very important to track. You can tell what phase you're at using a measurement called OD600, which stands for optical density at 600 nanometers, and this measures the light that's scattered by cells in your solution to quantify how many cells are present. So you can imagine that if a lot of light is scattered, it must be bouncing off a lot of cells. So you can conclude, okay, my media is very cell dense, that's a high OD600. So bacterial growth begins with what's called the lag phase, in which bacteria are first introduced to solution and they begin to metabolize and adjust, but they don't reproduce at very high amounts. So as a result, you'll see a slow increase in OD600. Eventually, you reach the log phase, where bacteria begin to reproduce more rapidly. They've become accustomed to their conditions, and now they're splitting. And since bacteria reproduce through binary fission, this is going to be an exponential relationship, so essentially 2 to the n. And your OD600 will increase very rapidly. Beyond this log phase is the stationary, which leads to the death phase, in which the media becomes nutrient deficient as those bacteria proliferate and begin consuming all of the nutrients. And so they start to die off due to overpopulation. And as a result, they release these toxic waste products and metabolites that then cause more bacteria to die. And the cycle repeats itself until the media has essentially become toxic for bacterial survival and it really can't support protein growth or protein production. So you'd want to catch the bacteria in the log phase because this is when they're most prolific and they're most metabolically active. And, if you wait too long and hit the stationary phase, the bacteria are going to be dying off and they won't be as good at producing proteins. So typically you can catch them at this phase by removing the flasks from the incubator at a certain optical density. And this value is dependent on the nutrient density of your media usually. So if you use a very nutrient dense media like TB, your bacteria can usually grow to higher optical densities because there's more nutrients to support them. Whereas if you use a more nutrient deficient media, you have to be much more careful about removing those flasks at a lower optical density to prevent them from reaching the stationary phase. And it's very easy from personal experience to overgrow your bacteria. So they need to be monitored very carefully, especially once you reach the log phase and they start growing so rapidly. So from there, you're going to cool them at 0 degrees Celsius to prevent overgrowth, so essentially halting this binary fission process. And then you induce gene expression by supplementing your media with a particular molecule called an inducer. And these inducers are designed to bind or activate the promoter that you've introduced into your plasmid. So way back when we were doing construct design, we created a plasmid that expresses our gene of interest in a switchable manner. So it can be switched on by what's called an upstream promoter, and that promoter will then recruit transcription factors and allow the transcription process to occur, which ultimately leads to your protein being pumped out. So one common operon that's used very often in synthetic biology is the lac operon, which binds and is activated by lactose. Usually experimentally, you'll use a lactose mimetic called IPTG, which serves the same exact function. It'll just bind to this promoter, it'll recruit transcription factors, and it'll allow the downstream transcription process to occur. Once you've added in this inducer, essentially switching on the expression of that gene of interest, you incubate your bacteria for a set amount of time, usually overnight at a lower temperature. Then you harvest your bacteria from the media by transferring your culture flask contents to a centrifuge tube, and centrifugation spins those bacteria around at a very high rotational velocity, which allows the denser material, so that's the bacteria, to kind of precipitate out and form a pellet, whereas your lighter media and everything that's soluble in the media, so that's your nutrients, your metabolites, that's going to all float to the top because it's less dense. So at this point, you have your bacteria in a pellet, and these contain your protein of interest at unusually high intracellular concentrations. But we run into another problem when we consider that bacteria, like all cells, must protect their contents from the external world using some kind of semi-permeable barrier. And in the case of gram-positive bacteria, that's a thick peptidoglycan-based cell wall in combination with an interior lipid membrane. And for gram-negative bacteria, that's an interior membrane, a thinner peptidoglycan cell wall, and then an external lipid membrane as well. So either way, to extract the contents of the bacteria, we need to break both the cell wall and at least one lipid membrane. And this can be difficult, so it's where the process of lysis comes in. Bacteria are typically lysed using two methods, which can sometimes be applied in combination and sometimes have to be done in isolation or selected very carefully to make sure that you're keeping the protein safe as you lyse and break open these bacteria. The first method is called mechanical lysis, where, as the name suggests, the cell walls and membranes are disrupted under intense mechanical stress. This is often done using sonication, where the pellet is suspended in some sort of stabilizing lysis buffer, and a sterile probe that emits high-frequency sound waves is submerged in that buffer. These sound waves provide enough energy to break apart the cell walls. The caveat is that they can also provide enough energy to heat up and denature your protein, so sonication has to be done carefully. You usually do it on ice and in short bursts to give the bacteria time to cool down and recover. Another common mechanical method is called French press, where bacteria are subjected to high pressure followed by rapid depressurization as they're pumped through a tiny valve, which creates shear stress that destroys the cell walls. Again, because this process can create a lot of heat, it must be done at ice-cold temperatures, and again in a stabilizing lysis buffer. Given that mechanical methods can be so intense and often so heat-intensive, chemical lysis is sometimes preferred for sensitive or easily denatured proteins. So in one chemical technique, the lysis buffer is supplemented with lysozyme. That's an enzyme that hydrolyzes the chemical bonds that make up bacterial's peptidoglycan cell walls, and that releases the cell contents. In other methods, you add in detergents, which will solubilize and break apart the lipid membranes, and other solutions and solvents can be used. You may have noticed that the theme of this Lysis section of the podcast has been about denaturation and protecting your proteins from denaturation. So denaturation is the process by which a protein loses its three-dimensional folding and can then aggregate or linearize or form all sorts of weird misfolded shapes. Because in biology, structure equals function, denaturation can be really harmful to the outcomes that we hope to achieve with protein engineering. So imagine we've engineered a protein to bind to a specific receptor. We want it to keep its three-dimensional structure that allows it to recognize that receptor. Because if it's misfolded or denatured, we lose that function. One very powerful denaturing agent is heat. So heat confers kinetic energy to the proteins, and that can actually provide enough energy to lift them out of their potential energy wells that represent their stably folded conformations and allow them to take on misfolded conformations. So proteins must be kept cool, and in my experience, you can usually get away with keeping them at 4 degrees Celsius, so either on ice or in a cold room, from the time that the large cultures are pelleted onward. Other chemical factors can also cause denaturation. Often this is in the form of excessively acidic or basic solutions, which can denature the proteins by either protonating or deprotonating certain amino acid residues to break ionic bonds that form between acidic and basic residues. So at physiological pH, most acidic residues, that refers to their side chains being acidic. These acidic residues are deprotonated, meaning they have a negative charge. And the basic residues are protonated, meaning they have a positive charge. And that allows them to form electrostatic interactions. But when you mess with the pH, all of a sudden you can end up deprotonating those basic ones or even protonating those acidic ones, and that'll break those ionic bonds by making them neutral. So it's critical to keep the pH stable, and you can usually do that by adding pH buffers to your lysis buffer or your subsequent purification buffers. Another factor that you need to consider is that most cells contain enzymes called proteases that break down proteins, and this is very biologically necessary because proteins are so ubiquitous that they can easily accumulate and aggregate and form structures that are harmful to cellular function. This is how a lot of neurodegenerative diseases can occur because proteins are misfolding and clumping together and they're not being properly degraded. So proteases are critical, but in our case, we don't want the bacteria to degrade the protein of interest, so we add in protease inhibitor tablets, which allows those proteins to remain protected and prevents the proteases from functioning catalytically. At this point, the lysis process is complete, and your buffer now contains all the lysed bacterial cell membranes and walls, as well as the various metabolites and macromolecules that were floating around in the cytoplasm. And we refer to this cellular soup as the crude lysate. This lysate can then be clarified using centrifugation, which again is when that soup is spun at a very high rotational speed in order to precipitate out the denser components like the cell walls and membranes, which are insoluble, while keeping proteins, nucleic acids, and other polar macromolecules suspended in the buffer. And this buffer is now referred to as the clarified lysate. So we've clarified it, we've precipitated out those insoluble components.
This goes hand in hand with another important topic, protein purification. Unfortunately, the bacteria aren't just going to hand us a perfect, completed protein with nothing attached to it.
At this point, your lysate contains a medley of bacterial proteins. And to separate your protein of interest, you need to have some sort of system with a high affinity for that protein in particular, and no other cellular proteins. This is a tall order, but we have a system called affinity chromatography, which really helps with the purification of specific proteins. And to understand affinity chromatography, we need to again go back to the plasmid and construct design process. When we encode this protein, we encode it with a certain tag at the very end or beginning of the protein, usually not in the middle because that'll disrupt its structure, that consists of certain unusual amino acid sequences. And these sequences have a high affinity for whatever stationary phase we're going to purify with. So one of the most common tags is the polyhistine tag, which consists of 6 to 10 histidine residues. Amino acids, or amino acid residues as they're called in polypeptides, are the building blocks of proteins. And while they all have similar backbones, they're all essentially the same except for one amino acid, proline, which has a twisted backbone. They have variable side chains, and the chemical properties of these side chains are what gives the proteins their unique folding and properties. Histidine has a very cool side chain called an imidazole ring, which contains a nitrogen atom that coordinates well to certain transition metals, such as nickel or cobalt.
Pause. Transition metals are simply specific metals found in the large middle block of the periodic table, and coordination/coordinating is simply one way in which atoms attach to each other.
Proteins that are labeled with many consecutive histidine residues, like our histidine-tagged protein of interest, will then have an unusually strong affinity for transition metal columns. So if we run our clarified lysate through a nickel or cobalt column, the histidine-tagged protein of interest will bind strongly to the column, while most other proteins that don't have this sequence of strong transition metal coordinating histidine residues, they'll flow straight through. There's always a possibility that another protein might bind weakly to the column through one of its histidine residues, or maybe through some kind of other coordinating atom, and this is why we follow up the initial flow through with a wash, which is when you basically flow another buffer through the column. And this buffer is typically rich in salt, which breaks electrostatic interactions, so positive, negative regions of different proteins can sometimes attract each other through, electrostatic Coulombic interactions. And the salt will break apart these ionic bonds. We also add a little tiny bit of imidazole to this buffer. And as you might recall, imidazole is that molecule that coordinates well to transition metals. And it'll coordinate so well that it'll kick off these weakly attached proteins. So proteins that are attached by maybe one or two histidine residues will be kicked off by the imidazole. But ideally, our protein of interest, which is attached by 6 to 10 histidine residues, won't come off as easily. Finally, we collect our protein by passing another buffer through the column called an elution buffer. And this contains a much higher concentration of imidazole. This imidazole has a high affinity for transition metals again, and since it's present at such a high concentration, it'll displace our protein, allowing it to flow through in a process called elution. And sometimes you'll find that your buffer isn't strong enough to elute your protein fully. That's when you'll scale up your imidazole concentration to allow that molecule to successfully kick off all your protein. Now, we can actually encode some kind of protease recognition site into our amino acid sequence, where those proteases that cut apart protein will cut the polyhistidine or the affinity tag off the protein, leaving only the native structure. And this can allow for a lot of structural characterizations like cryo-EM, which require the protein to have only its native amino acid sequence with no appendages or tags hanging off of it. Although affinity chromatography is typically a helpful first step, it can be, and often should be, followed by other separation techniques to improve sample purity. Size exclusion chromatography is a helpful follow-up usually, and it separates proteins based on their size, which is especially helpful if your protein tends to aggregate or form oligomers or dimers, and you only want the monomer. In this chromatography technique, your lysate is passed straight through a porous column, and because this column contains pores that smaller proteins can stick in, but allows larger proteins to come straight out, you're going to have the proteins come out in a gradient that's inversely related to size, with the large proteins flowing straight through and the small proteins kind of taking longer to get out of the column. This is an inverse relationship that's completely different from what you would expect in other separations of gel electrophoresis, where the small proteins or small DNA fragments can make their way much more easily through the gel matrix, and so you have small protein bottom, large proteins on top. In size exclusion chromatography, it's large proteins out first, small proteins out last. Another common separation technique is ion exchange chromatography, where your affinity output is passed through a column of charged resin. Similar or neutrally charged proteins will pass right through because they don't have an electrostatic attraction, or they might even be repelled from the column, whereas oppositely charged proteins are going to be retained by Coulombic attraction. This process is a little bit tricky because it doesn't distinguish between similarly charged proteins. So in your lysate, you'll expect to have a lot of bacterial proteins that are positively charged or negatively charged. And so it can be challenging to put an enormous mixture of proteins like your initial crude or clarified lysate through the column, because many proteins will be retained and they'll contaminate your product. However, it can be helpful in certain cases, like if you have a DNA binding protein, that's often a positively charged protein, that's been purified, but it continues to stick to loose bacterial DNA through electrostatic attractions. The ion exchange column can help break up these interactions and allow you to elute your protein in high purity.
If you've been in a lab or you've been following the podcast, you probably know what's next. testing what we did to ensure success. Let's cover SDS-PAGE again, one of the most common protein analysis techniques you'll see in basically every iGEM lab. The idea behind SDS-PAGE is pretty elegant. You take your protein samples and mix them with SDS, which is a detergent that unfolds proteins and coats them with a uniform negative charge. As we've mentioned before, negative charges will move towards positive charges. So, when you run them through a gel under an electric field, the proteins separate primarily by size. Smaller proteins move faster through the gel matrix and larger ones lag behind. Once the run is finished, you stain the gel so the protein bands become visible. Ideally, you're looking for a band appearing at the molecular weight you predicted for your construct. You can check out our last episode where Richard talks a little bit more about it. But SDS-PAGE only tells you so much. A band at the right size doesn't necessarily confirm identity. Lots of proteins can migrate similarly. That's where Western blotting becomes really useful. A Western blot basically takes the separated proteins from the gel and transfers them onto a membrane, usually made of nitrocellulose or PVDF. Then, you probe that membrane with antibodies that specifically recognize your protein. The antibody step is where the specificity comes from. Antibodies are proteins naturally used by the immune system to recognize and attach to specific targets, like viruses or bacteria. Typically, you start with a primary antibody that binds specifically to your target protein. Then, a secondary antibody binds to the primary one and carries some sort of detectable label, often linked to an enzyme that will light up during a reaction. We take advantage of that property to identify if our protein is there. If it lights up, then it's there. Another important aspect to this testing process is enzyme assays and functional testing. Let's hear a bit about that from George Zhang, one of our full team leads this year.
So an assay is basically just any test where you can determine the effect of the protein, whether it's through like the formation of a product or a color change through a colorimetric assay. There's a whole suite of assays and tests that you can use, while functional tests refer to just the action of testing the function and the catalytic effect of an enzyme. So one thing we want to look for is enzyme activity. Basically, all enzymes are able to catalyze or help chemical reactions that occur in the body or in the cell. And for example, going back to the 2024 iGEM case, we worked with perchlorate reductase. So that's an example of a assay, right? We have the perchlorate reductase enzyme in like a system. We add the compound that the enzyme affects, in this case perchlorates. And with the two, we're then able to measure the degradation of perchlorates, which is a direct measure of how well the enzyme is working. And we're able to quantify the activity of perchlorate reductase. That's just one example though. Like there are other examples where it's like colorimetric assays. So for example, let's say you had a protein or enzyme, and what it did was it broke down this compound, and the new compound actually caused the color of the solution to change, right? That's another way to assess the activity of the protein. And what's interesting is when you have controls, when you bring in experimental designs, right? Now you'll have multiple variants of the same protein, or you can have like controls with like no proteins, and you can kind of assess the enzyme activity. One thing that's interesting is enzyme assays and functional testing is tied with this idea of adaptive evolution. So what this means is you'll have proteins, and the sequence of the proteins is kind of already set, and you're able to test the activity through an assay. Now, what if we wanted to improve the function of these proteins, right? Like we have the assays and we can determine how fast the protein acts, and we can determine how specific the protein is. What if we want to improve the protein, right? What if we want to improve its catalytic activity? And we need assays to do that, because those assays determine like the baseline performance of the protein. And then you can run experiments where you adaptively evolve the protein and the corresponding DNA sequence, which codes for the protein, and you can obtain better variants of this protein, and you assess the betterness of this protein by running assays, right? You'll have your starting protein, and you can compare that activity to your modified protein through adaptive evolution. So for example, the 2023 iGEM team here at Cornell, they had an adaptive evolution experiment where you need a driver to affect like mutations to create new variants, and their method of doing that was performing error-prone PCR. So they took a existing DNA sequence which would code for a protein, and they ran it through a PCR machine where this PCR mixture had a deficiency, and it went in magnesium chloride concentration. And because magnesium ions are necessary for polymerase activity, the reduction of the magnesium ions resulted in mutations that occurred in the resulting DNA transcripts. And because of that, we're now able to run an adaptive evolution experiment where you have these new variants of the protein sequence in DNA form, and you express them in cells. In their case, they were trying to break down caffeine into methylxylazine, which had potential to treat myopia. And they had a selective pressure to basically screen out the bad proteins, right? The proteins where the mutations were actually not beneficial, and it reduced the efficiency of the protein. And what happened was they were able to obtain new variants of the protein which were more efficient than their starting variant because of this adaptive evolution and because it was guided by enzyme assays and functional testing. So that was a lot. Just to summarize, so enzyme assays, functional testing, they're used to determine how good your protein is currently. And once you're able to assess how good a protein is, you're now able to begin creating improvements in that protein, right? You're able to now maybe improve its efficiency. You're able to improve its substrate specificity so that it only binds and affects one protein or one compound, and it doesn't have any side reactions and side effects. And you can do this, for example, through this process I described called adaptive evolution, where you go and you purposely create mutations in the protein and you kind of screen the activity of these proteins through assays, and you keep the best proteins. And again, it's called adaptive evolution, right? You're keeping the fittest proteins, and you're constantly adapting and affecting mutations to kind of drive that change. So that's a really cool application of, once you have your proteins and your enzymes, putting enzyme assays and functional testing to good use by trying to make their performance better. There's also other things you can do with enzyme assays and functional testing, because it's also going to be a function of how well the cell is producing these proteins and enzymes. And that's where metabolic engineering comes in, where this is going to be the last application of enzyme assays I'm going to describe. But there's so many different assays, and there's a lot of creative ways you can come up with enzyme activity. But going back to metabolic engineering, protein activity is going to be a function of how much enzyme you have in the solution, and that's going to be a function of how well the cell is able to produce this protein. And we had this issue in 2025 where we weren't able to produce enough of the perchlorate reductase because it was a very complex protein. It had 4 subunits. So it was actually very hard for us to produce the compound and enzyme in high concentrations. But you need that if you're going to run assays and enzymes. So that's where metabolic engineering comes in, where you can actually modify the metabolism of the cells of interest that are producing these proteins, and you can make the production of your desired enzyme or protein more desirable, and you can artificially improve the yield of your desired product through this process. This was done in 2024 iGEM, where we studied the metabolism of yeast cells, and we actually knocked out a pathway and process to kind of shunt more starting material to our desired protein. But going back to enzyme assays, I'm going to give an example of the most interesting assay I've seen. And when I've kind of described assays, you've probably been thinking of you need calorimetric, you need compounds, you need products and whatnot. It has to be very wet labby, right? But that's not the case. So this team actually won. They won all of Jamboree. They're a crazy team, and they also won this thing called best measurement. So enzyme assays and functional testing are tied to the gold medal best measurement at iGEM Jamboree, where it's as the name suggests, right? You're measuring what the protein is doing. And the team that does it best gets the best measurement award. And this team, you won't believe what they did. So they were trying to prevent forest fires, and they did that by creating a gel. It was very flame retardant, this gel, and it could be sprayed on trees and bushes and forests, and it would be degradable, right? It was very safe for the environment and it would prevent fires. And what they did to test this gel was they literally applied the gel to cardboard and they burned the cardboard. And it was such a creative way of measuring the effectiveness of their proteins and their enzymes. It was in a way an assay because it was measuring the activity of their construct. That they won best measurement, because come on, they're literally burning stuff in lab. That's one of the craziest experimental designs I've ever seen. And it's crazy how creative they were. But it goes to show best measurement and any gold medal in iGEM. There's so much room for creativity and imagination, and iGEM really does reward that.
Every purified protein gives scientists a clearer view of how cells function, how diseases develop, and how new medicines can be designed. Many of the advances we rely on today begin with the ability to isolate a single molecule from the busy mess of the world. We went through a lot in this episode, so thank you for sticking with it. If you have any questions, make sure to leave a comment or ask your favorite scientist, even if that scientist is just Google. In the next episode, we're going to be applying all the techniques we've talked about so far in organisms. Until then, stay smart, and I'll see you geniuses soon.